Hello students, welcome back to my class. So here I am with the new video and new topic. So dear students, if you remember in my last class I had taught you the chromosomal theory of inheritance. So I hope that you all have watched my last class and you all have written notes on it, and I also hope that you have understood the topic very clearly. So dear students, so today let us start a new topic, that is linkage and recombination. So most of the students, they find this topic very difficult, and the reason behind this is the understanding, okay, of the terms, that there are few terms related with this topic which is not clear, okay, to students. So for that reason they were not able to grab the concept, they were not able to follow the concept. So before I start this, so let us start one by one, okay, so that in a sequence, so that you all can follow me and you can understand the topic clearly. So before I jump into the what exactly the linkage and the recombination is, let's start with independent assortment. Okay, so in independent assortment you have already learned this twice, right? I have already explained this in chromosomal theory also. I had explained it to you, right? Okay, so I am not okay. So let me start with independent assortment. Suppose there is chromosome, right? And this is A, capital A, and this is small a, okay, and there is another, this is again capital B. See, in independent assortment, if you remember, there two contrasting characters were taken, right? That's why I'm making different, okay, this is one and this is another, and this is represented with small letter. Now what happened? Case 1, okay, case 1, what are the possibilities? See, I will be showing you the possibilities. Now the possibilities is, okay, or you can say the possibility 1, what could happen? Okay, just listen here. See, whenever the linkage, whenever there is crossing, okay, when they mate with each other, the possibilities either this will pair up with this, correct? I hope you all can follow. This capital letter A means both the dominant characters may come together, right? Okay. Second thing, or you can say, okay, second possibility is that either capital A can pair up with the recessive, means the dominant character may pair up with the recessive character. Capital A, this is small b, correct? This is case 2. Then another probability that maybe both the recessive character may come together, right? So maybe both the recessive come together. This is case 3. This is, or you can see the possibility 3. Then the fourth one is either one dominant and one recessive. That is, this may pair up with this. The possibility is this, okay, either with this or with this. I hope you all can follow me. Okay, so okay, now here you see independent assortment, right? According to this, dominant should have paired up with dominant, right? Right, only this could have happened, but here you can see that, or dominant and recessive, and the result will be a dominant, but here you can see the variation, right? So from here what we can conclude is that, as I'm repeating the independent assortment, that the this alleles, these gametes are not dependent, the characters, the traits are not dependent on the other trait, okay, they're independent of each other when they segregate, right? So this is called your, what does it call? It calls your this is called your independent assortment. Now what happened in a chromosome? See this, see when they separate, okay, what happened? This are together, right? They will go together. So what happened when you unwind it, or when they get separated, here you will see that this is one gene A, B, A, B. Could you say, I'm just taking this example, okay? When they just get separated, so what happened in a one chromosome, this is gene A, okay, and this is gene B, right? So here you can see that they are together, right? Could you follow me? Now, these two genes are on same chromosome. See, this is the same chromosome. They are not different. Okay, this is same chromosome. And this phenomenon, okay, this phenomenon is called your linkage. And this gene, which are at the same chromosome, this gene are called your linked genes. Could you follow me? Why they are linked? Because they are on same chromosome. Clear? And this is linkage because they are linked together. Linkage means what? Having some connection, right? To have some connection, or the connection between any things, okay, between any two is called your linkage. And this gene are called linked gene. I hope that independent assortment, what is linked gene, what is linkage is clear, right? Okay, then what happens? Now, how does this happen? Now, the question is, how does this happen? Okay. So, what happens is, suppose there is chromosome, okay? This is A, this is dominant A, this is dominant B. Okay. Now, let us take another example with dominant and recessive character. This is recessive. Okay. See, here what happened? They just cross over. Okay. What happens in them? The things, okay, the chromosome, they just cross over one over another. Okay. In crossover, because you can see that the sign it makes like cross, right? Okay. So, because of this, what happens? See, you can see that this two, okay, they are what? They are just crossed over. So, as a result, what changes you will get to see is that this part, this gene A will come over this, and this gene A will come over this, because they are crossing over, isn't it? So, this will be your A and this is your B. And here you see this is your small letter B and this is capital A. Could you see this? And this is called your, this method, that how it happened, okay, this process is a crossing over and the actual result. So, this is a recombination. So, recombination is done by crossing over. So, see, this is method that they crossed over, okay. Recombination means they're recombining. Here you see the pair. It is capital A, capital means both are dominant, right, and both are recessive character are here. Am I clear? But here, when this crossover takes place, when crossing over takes place, there is an exchange of gene. The genes, they were exchanged. Could you see this? So, this part has been exchanged. It means what? They are recombining with each other. There is a recombination. Combination means what? To bring together, to combine, right? Re means again. So, they are combining again. Earlier, this was the original one. Earlier, it was like this. Because of crossover, because they crossed over, the chromosome crossed over with each other, that results into recombination. And this gene, as I told you, these are called what? These are called linked gene, and the phenomenon that they are linked with each other, linking, okay, that is called your linkage. So, I hope that this is clear to you. What is linkage? What is linked gene? What is recombination? So, if you have any doubts on this, then you can ask me, okay? Now, there are few things. So, on this, what happened? Okay, after this, they will produce gamete. Okay, So 4 type of gamete will be produced. Now what are those 4 type of gamete? AB, correct? A by B, and small letter A, small letter B. Okay, so as you know that when a gamete form, the 4 gametes are formed. Now one thing you should remember is that linkage, okay, this recombination, whether this recombination will take place or not, it depends on the distance between the linked gene. Okay, here you see, if the distance between 2 gene, when they will cross over, there is a less chance that this chromosome, okay, they will get to that, okay, they will get exchange. Are you clear? So if the distance is more between this, then there will be less chances of linkage. Sorry, there will be less chances of recombination, less chances of crossing over. Could you follow me? That if the distance between the gene on the chromosome, okay, if it is more, the distance is greater, then there will be less chances of recombination, okay, and if the distance between them is less, then there is maximum chances of recombination. So remember, this type of questions comes in your entrance exam as well. Okay, all right. So in this way we can say that the linkage, okay, it is inversely proportional. Okay, it is inversely proportional to the distance of its gene, correct? And recombination is directly proportional. Am I clear? Linkage is indirectly proportional. If more distance, less linkage. Okay, are you getting it? So this is linkage, right? The phenomenon. And the distance is less, it means more linkage. If distance is more, then less linkage. Okay, now how does this linkage happen? So in that in that topic, with base on this, what happened? An experiment has been given, okay, in your NCERT book, and it was known as T.H. Morgan experiment. So it was given by T.H. Morgan. So what he did, he chose Drosophila melanogester. See, the way Mandel did the experiment on plant, right? He took a pea plant because it was easily available, it can reproduce easily, right? The life cycle is short. So in the same way, T.H. Morgan, like, he did experiment on animal because experiment was already done on plant. So he chose Drosophila melanogester, which is a fruit fly. Now why does he chose this? So Drosophila melanogester, he chose this for some reason, okay. First is that variation in trait. The way pea plant has so many variations, so many traits it has. Color, shape, isn't it? Height, there were so many. Similarly, T.H. Morgan, he chose Drosophila melanogester because of the contrasting trait, like eye color, red and white. So Drosophila were found with 2 eye color, red and white. Then body color, that is a brown and yellow, and wing is miniature or large. As well as these were about the traits, that these were the trait. It's eye color, body color and wing, right? So eye color, red and white, remember this. Body color is brown and yellow, and wing is miniature or large. As well as 2 weeks life cycle. So their life cycle was 2 weeks. So it was easy to reproduce. It was easy to grow, okay, in lab as well. They can easily grow them in a lab as well as, and it was easily available because the life cycle is only 2 weeks. So these were some characters, okay, of Drosophila, for which T.H. Morgan chose Drosophila for his experiment. Now, so here you can see the symbol taken, okay, like for dominant characters, they were called as wild type as well, okay. Now in Drosophila, brown body, okay, Drosophila with brown body were wild type, means they were dominant characters. So those who were dominant were represented with this plus sign, okay. So brown body means Y plus, Y positive. If it is yellow body, then Y. It means this is dominant, this is wild type, and this is recessive. So yellow body flies were recessive, okay. Red eye, it was represented as W positive, and this is your W. Okay, and for the wings, okay, if the wings were large, then it is M positive, and if it is miniature, okay, means the small recessive, then it is only written as M. So these were the symbols, these were the characters of the Drosophila melanogaster. I hope that you all can follow. Remember that if there is plus sign, it means their dominant character. Clear? And if there is no sign, simply written, then it is what? It is your recessive character. Okay. Now what happened? T.H. Morgan, okay, he did an experiment first with this brown body and with the color of the body and with the color of the eye color and body color. So let us see the experiment that he carried out, okay, and do listen this carefully. This will help you to understand.